My name's Rebecca Parr, and my talk today is a new antiviral therapeutic for the prevention of rotavirus infections. Now, rotavirus is a double-stranded, segmented RNA virus in the family of Virology. It commonly infects infants and young children and causes a severe diarrhea and dehydration. The symptoms include decreased urination, dry mouth and throat, dizziness, watery diarrhea, vomiting, and fever. Now, we have two good vaccines here in the United States that are licensed, but they do not completely protect. They protect from severe diarrhea, but you still have the virus growing and replicating, but you don't have the pathology. And another thing, and there's another vaccine that is now licensed in India that's very good and very effective in India. But these vaccines are strain specific. And as y'all heard recently, there are some other vaccines that different companies are working on as we speak. Diarrhea treatment, we can treat the diarrhea with oral rehydration salts, with zinc supplements. Intravenous fluids is the usual thing that we use for the children, and we get nutrients in these children. We need some very rich foods, but we have no antiviral drugs for rotavirus. So I was at Arkansas State and listening to a friend of mine give a talk, and he's really excited about medicinal plants, and he takes the roots from peanuts and he immortalizes them and stimulates them to think that they are being infected by a pathogen. And they produce these very small molecules in the supernatant. He takes the supernatant, highly purifies these, and what he's found, and he characterizes these, and he's found that he commonly can find different stilbenoids that are produced by these roots. One of the most common stilbenoids that most of y'all might have heard of is Resveratrol, that's also found in the skin of grapes. It has biological activities of being antioxidant. It has anti-inflammatory, cardioprotective, and antiviral and anti-cancer and anti-aging properties. Lots of different properties. But the problem is that some of these, and they're all hydrophobic as you can see, but some of these have limited oral bioavailability, and this possibly is due to the rapid absorption and metabolism. Now, two of the compounds, resveratrol and picynitol, which just for the sake of saying that word, I'm going to call it TPA from now on. We have some compounds that are also prenylated. They look very much like their parent compound. Like their parent compounds, except they have the prenylation on the end, and that gives them some different bioactivity that we're going to talk about in just a few minutes. The first thing I had to do was look at my cells and try the 4 different stilbenoids that I had and make sure with all the different treatments that these were not affecting the viability of the cells. So I checked all of these, and I've done these numerous times, and there is no statistical difference between our treatment and the untreated cells. So then we were able to go further with our studies, and we took the different stilbenoids and we put them with the virus in our cell culture for 1 hour during the infection. Took them off, and then I had 2 sets of experiments where I collected the cells and supernatants at 12 hours post-infection and 24 hours post-infection. I took the supernatants and looked for infectious virus particles, and I took the cell lysates, and I'll show you some data in a minute where we look for viral proteins. So the first one, looking for infectious virus particles, I did focus forming units and found, this isn't working. But I found at 12 hours post-infection, I saw no statistical difference between any of the treatments. And at 24 hours post-infection, I didn't find any statistical difference between the resveratrol and the TPA. But I did see statistical differences between the TA3 and the TA1, the prenylated forms of these stilbenoids, and they were statistically different. I then moved to Stephen F. Austin State University, where I started up my laboratory, and was not able to do the focus-forming units. I didn't have the antibodies to do that, so we went back to using the tried and true plaque assays on the same supernatants to show could we get the same results or similar results. And at 24 hours post-infection, we saw very similar results where the resveratrol and the TPA show no statistical difference between treatment and no treatment. But I saw beautiful data of TA1 and TA3. We did have statistical differences between the treated and untreated group. So we are seeing differences in the production of infectious virus particles. The next thing we wanted to do with the cell lysates was to look at viral protein, and a specific viral protein called non-structural protein 4 that's very important for viral replication. And with this, we added equal amounts of our cell lysates from our experiments to an SDS page gel, ran it, blotted it onto nitrocellulose, and did our Western blot analysis. And as you can see, we have a tremendous lower amount of NSP4 in our two treatments, TA1 and TA3, than we do from non-treatment or treatment with just DMSO. So we have a difference in the infectious virus particles that we get from the system, and we also are having an effect on virus replication. And that's very important to know those two. So at that time, my friend, we're starting to try to figure out what is the mechanism of action, how is this working? And my collaborator at Arkansas State had done another study with another group, and they found that these two stilbenoids actually bind to cannabinoid receptors 1 and 2. And he asked, do you have these receptors on your cells? And I went, I don't know. So I did slot blot analysis on both the human intestinal cell line that we used and the African green monkey kidney cell line that we use for plaque assays. And I happened to have some infected cell lysates also with that. So it was very nice to see between uninfected and infected, there seemed to be an upregulation of both these receptors. So what are these cannabinoid receptors 1 and 2? Well, they're classical cognate receptors for all types of agonists. And they are the cannabinoid receptor 1 you can find in the brain. You can find the cannabinoid receptor 2s on some immune cells and in different tissues of the immune system. And it's also, both of these are distributed throughout the GI tract, predominantly in the enteric nervous system. And that gave me good thought to think this may be important for us. Another nice thing in the literature is both these receptors are upregulated during intestinal inflammation, and that's exactly what happens with a rotavirus infection. So that made us feel comfortable. And then we wanted to find out, how do they show their effect? What's their mechanisms? Well, cannabinoid receptors have been shown to have several different apoptotic actions on different cancer cell lines. And the cannabinoid receptor 1 mediates its effects through decreasing the RAS MAP kinase. Oh, thank you, appreciate it. The MAP kinase and PI3 pathway, and it decreases cyclic AMP and the PKA pathway. At the same time, we're having an increase in the ceramides, which all 4 of these lead to apoptosis. So that made us think, let's look at the apoptotic pathway and see what we can find. And one of the laboratories next to us was working on an apoptotic pathway project, and they were very kind and gave us their primers to do quantitative PCR and see if any of these caspases were important in our system. And what we found here is that looking at caspase 3, 7, 8, 9, and 10, we see some variations and a little bit more than a variation on caspase 9, but really there were no statistical differences with the others. But this is at 8 hours post-infection, and rotavirus doesn't kill our cells in culture till 18 hours post-infection. In the literature, caspase 3, which we have right here, is the one that appears to be important with rotavirus infections and the leading up to apoptosis. But at this early time point, we don't see anything here. So what we have to do now is do our time course and look at the different transcripts up to 18 hours. And that's what my students are doing as we speak. Another nice thing I need to tell you is that we've switched from using the simian rotavirus, SA11-4F, to using the human rotavirus, which is much more applicable to what we're looking at. And we've repeated a lot of these different assays with those, and we're seeing very similar results. But the numbers of virus particles are so much higher, which has allowed us to use a new technology, the Q nano system from ISON, which we can actually count virus particles, and we can look at the relative sizes of them and see what the population looks like. And with that, with one of our, this is just an example of looking at TA3, and we actually have measured about 9 times 10 to the 11th virus particles per ml. So this is giving us a new tool to look at not just infectious virus particles, but a variety of the infectious and non-infectious virus particles and the different sizes. Along with looking at the transcripts, I have a student that wanted to look at the proteome. So she has started doing 2D gel analysis, and she's worked through the different stages of working through getting her assays to look good, where we can actually compare them. And she's getting ready to repeat these experiments and getting into the numbers so that she can analyze them. So with that, I'd like to acknowledge my lab, my 2 master's students, Hannah Lockwood, Caleb Witcher, my 2 undergraduates, Raven Slay, Macy Mattia, and Ron Havner, who helps us with things as they get broken or need to be fixed. He's an excellent help. And Dr. Judith Ball at Texas A&M, who is helping us with the virus, and our collaborator, Fabrizio Medina Bolivar at Arkansas State. Thank you. Do we have any questions?